Okay, well, thank you for that introduction, Carrie, and I'm happy to be here and talk with you a little bit about what we're doing at Kansas State University to look at alternatives to antimicrobials using the gut microbiome. So we'll start out with the basics. The definition of the microbiome is the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. But I think even more so we can simplify that into symbiotic, or those microorganisms that provide us benefits, and pathogenic microorganisms are those that provide us harm. So if we consider the pathogenic microbes to be these and symbiotic to be these, our natural instinct as a veterinarian or a practitioner is to eliminate the pathogen. So whether that be through creating a vaccine or prescribing antimicrobials, the focus is, of course, the pathogen. Largely ignored the beneficial microbes. And so when we think about the microbiome as an alternative, ultimately what we're trying to do is, can we get the same effect by increasing the beneficial microbes? So can we either reduce the effects of the pathogen or actually eliminate the pathogen itself just by increasing those beneficial microbes in the gut? So there's lots of things that impact the microbiome development in just the first 3 weeks of a piglet. These initial characteristics have to do with the sow, so whether that be the vaginal microbiome, the fecal microbiome, and even the skin microbiome as that piglet is nursing. Additionally, we know that the environment plays a major role, so if that pig is housed outdoors versus inside, is it a filtered barn versus not? And of course, the transition to diet plays a major role in the microbiome colonization. We know that stress at processing through needle teeth clipping and tail docking, as well as, of course, injectable antibiotics, have the potential to impact the microbiome for actually the lifetime of that production animal. And then, of course, pathogens as they're exposed and in-feed antibiotic growth promoters. So what's important here is that all of these factors can occur just within the first 3 weeks of a pig's life, and it provides us with lots of opportunities where interventions can either provide benefits to the microbiota or we could provide detrimental effects to the microbiota for the lifetime of that pig. So there's really three main roles we think about with the microbiome. First, we know that those gastrointestinal microbes are essential for digestion and absorption of nutrients. We also know that the microbiome plays a protective barrier function. It's termed more of a colonization resistance to those pathogenic microorganisms once they come into contact with the gut. And then we also know, importantly, in the health of the animal, the microbiome plays a major role in both the development and regulation of immunity. So in my laboratory, we focus on respiratory disease. And to this audience, it shouldn't be a surprise because respiratory disease, infectious respiratory disease, is one of the major causes of antimicrobial usage in swine and cattle. And it's arguably one of the highest health challenges to pigs and cattle in the industry. And so when we think about how the respiratory tract actually interacts with the gut, it's primarily through those products of microbial metabolism, but also through that stimulation of systemic immunity. We know that in the human literature there are several respiratory pathogens that have associations with the gut microbiome and outcome following respiratory infection. And so in my laboratory, we've really tried to understand how does that translate into a porcine model. We know that the microbiome impacts weight gain and immunity, and it impacts respiratory disease in humans. So how does that translate into our porcine patients? To simulate porcine respiratory disease complex, this is a very as the term acknowledges, a complex that includes viruses, bacteria, environmental factors, stress, transportation. And so we simulate that through using a co-infection model using two viruses, porcine reproductive and respiratory syndrome virus and porcine circovirus type 2. Importantly, PRRS virus is the most costly disease of swine production worldwide. It's been around for about 3 decades. There's been millions of dollars put into vaccine research, and it continues to be the most costly disease of swine. And so it's one of those diseases where a lot of alternatives are coming into play. So how can we alternatively look at this disease and improve outcome in opposition to, let's say, another vaccine development? And so these two viruses work together to actually enhance disease. They have primarily lung and lymphoid pathology. They reduce weight gain. They cause systemic wasting, so systemic infections, but again, primarily respiratory clinical outcome. So the first question that we wanted to ask was, just does this translate into a pig model? Are there associations with the microbiome with outcome following co-infection with these two viruses? And the first paper that we published showed that yes, there are microbiome associations, specifically increased diversity and the presence of a non-pathogenic E. coli was associated with improved clinical outcome 70 days post-infection. And the clinical outcome was defined as a complete lack of clinical signs after co-infection and the highest average daily gain within the pigs that we looked at. This led us to the question, are there actual microbiome characteristics that may predispose or predict growth? And so what we looked at in this second manuscript was, are there microbiome characteristics present at the time of virus exposure or before virus exposure that may predict or predispose outcome? And we found several characteristics that were associated with high growth, subsequent high growth after co-infection, including increased diversity, increased streptococcus, and increased ruminococcus. So after we established that, yes, there are microbiome characteristics associated with outcome, we started thinking about, okay, how can we prophylactically modulate the microbiome to sort of set this pig up for better response? And so, of course, this is the way that we use vaccines to stimulate immunity. And can we use the gut microbiome in a similar way to actually improve subsequent outcome? When I started thinking about how we could modulate the microbiome, I started looking at fecal microbiota transplantation. It's used in humans, and it's actually the process by which feces is collected from a, quote, healthy donor and subsequently transplanted into a young or diseased individual. In humans, it's only used in diseased individuals. Of course, in our cases, we used it in young individuals to sort of set them up for improved response. The way that fecal microbiota transplantation is efficacious is largely unknown. The mechanisms are not specifically worked out for most diseases. However, we believe that it works because it increases those beneficial microbes, increases diversity, and modulates the systemic immunity. So we looked at this using 23-week-old littermates. So we split them into pairs, and there was a control group as well as a fecal transplant group. The fecal transplant was actually donated from 2 sows with several high health characteristics, and it was transplanted for 7 days prior to co-infection and then compared with a control group, which was administered a saline mock transplant. We followed the pigs for 42 days after co-infection to evaluate what the transplant did to outcome in these pigs. Interestingly, the transplant provided a significant protection against clinical disease that occurred in the second half of the co-infection period, shown here, that actually resulted in a 60% reduction in antimicrobials. As part of our IACUC, the attending veterinarian, of course, prescribes antimicrobials as well as anti-inflammatory medications as pigs develop respiratory disease, similar to what we would do in the field. And what we saw was, again, the significant reduction in clinical signs and significant reduction in parenteral therapy. When we looked at the growth rate of those pigs, we also saw a significant benefit associated with fecal microbial transplantation. The control group, 70% of the pigs started to lose weight during the co-infection period and actually resulted in mortalities, whereas the transplanted group only had 20% mortality. Again, significant improvement in clinical outcome by using this fecal microbial transplant. We also found that, interestingly, the lung lesions were less severe in the transplanted pigs, and that there was an enhanced antibody response in the transplanted pigs, indicating that that transplant modulated systemic immunity, increasing antibody production and creating a more sustained antibody response for both viruses. So through our work so far, again, looking at the microbiome as an alternative to reduce antimicrobial treatment and improve growth and health in swine, we found several characteristics using this porcine respiratory disease complex model as far as clinical outcome, such as reduced clinical disease or increased growth, in association with several microbiome characteristics. But however, the exact mechanism by which the microbiome impacts clinical disease outcome is, again, relatively unknown, and these are, again, associations as opposed to causation. But this research is definitely a growing area and provides us with an area where we can benefit the overall health and growth of the pig. If we focus on fecal transplantation and the subsequent reduction in antimicrobials, it's interesting to think about what diseases are used for, in which fecal microbial transplant is used for treatment. So the only approved indication is recurrent Clostridium difficile infection that is non-responsive to vancomycin, but all of the diseases that are highlighted here can be diseases that are typically treated with antimicrobials but have shown a beneficial effect when that patient is treated with a fecal microbial transplant. Interestingly, there's also been some anecdotal evidence showing that in patients colonized with antibiotic-resistant organisms, fecal microbial transplantation can actually reduce those antimicrobial resistance genes and actually resolve those antimicrobial resistant infections. So when we think about how this has been used in livestock, in cattle, it's called transfaunation. Again, in other species, it's called fecal microbial transplantation. But how it's been used, it's actually been used in several livestock species for several different conditions. And what's interesting is that fecal microbial transplantation also has had a broad range of benefits in these livestock species, from everything reduced lung lesions, reduced diarrhea, increased feed intake, greater milk production. So again, we can see this broad benefit from the gastrointestinal microbes to the overall health of the animal. So there's several potential applications for this. The first one is going to be the one we're most familiar with. So this is what's used in humans where they have clinical signs of disease, and then the transplant is used to resolve those clinical signs. That could also be used in livestock. In pigs, it's also used as an immunogenic tool. So in gilts or sows, sometimes they are transplanted with feces that have enteric viruses to actually stimulate mucosal immunity, with the ultimate goal to increase antibody delivery to the neonate. And then what I think is probably the most exciting and how we used it here is the prophylactic use. So increasing the beneficial microbes in those animals prior to a high stress or a high risk time period in their life, such as weaning in piglets. These are the funding source, and again, thank you so much for this opportunity.